Hello, everyone. My name is Larry, member of the A+ team. My team and I will be presenting our research journal titled The Effects of Cigarette Smoking on Breath Holding Time in Healthy Adults, ages 18 to 40. But first, let me introduce the rest of my team members and what part they're going to be presenting. First is Mika, who will be presenting the last half of the introduction. Next is Marjorie, who will be covering the methodology part of the study. And then GM, who will discuss the result of our study. And finally, Mitra and Justin, who will tag teaming the discussion part. Next slide, please. Our primary goal is to understand if cigarette smoking has significant effect on one's ability to hold his or her breath. My team hypothesized that non-smokers can hold their breath for longer periods of time on average than smokers. Why are we concerned about breath holding? Because breath holding technique has some benefits, especially for athletes like swimmers who swim faster, and for runners or other athletes who play sports that require them to run faster, like sprinters, football players, basketball players, and so on. It also improves lung function. The longer they can hold their breath, the more they can tolerate strong feelings of breathlessness and improve their performance. Next slide, please. In preparation for our study, we looked at multiple studies on both relationship between breath holding duration and smoking, as well as studies examining the effects of cigarette smoking on lung physiology. And we were able to find these three studies: a study of Suda, a study of Budami, a study of Aditya. Next slide, please. In study with Suda, they used three different types of subjects: smokers, ex-smokers, and non-smokers. As you can see on the slide, they define each of those subjects. And also the age group that they use, both males and females. Also, as you can see on the result, as far as average mean of BHT of each group, statistical analysis showed no significant difference between ex-smokers and non-smokers, but shows higher mean BHT compared to smokers. In this study, they concluded that BHT is significantly lower in smokers compared to the other two groups. Go ahead, Mika.
So another study by Badami et al. Found that in men aged 20 to 50 years, a significant improvement in breath hold time was shown among non-smokers when compared to smokers. This study was conducted on a cohort of 94 men, excluding female subjects, as well as any subject who was exposed to dust and or fumes on a regular basis due to their occupation, as well as those who suffered from underlying illnesses or allergic disorders affecting respiration. Next. And so for the Badami et al. Study, smokers were defined as those who smoked 5 or more cigarettes per day for a year or more. And they had a mean breath hold time of 34.85 seconds. Non-smokers were defined as those with no history of smoking, and their mean breath hold time was found to be 46.61 seconds, which is an improvement of 11.76 seconds over the smokers. Next. And so our hypothesis is based on the assumption that smokers are more likely to have lung tissue that is damaged in comparison to non-smokers, and that the damage in lungs of smokers may be such that they offer a less effective surface for gas exchange. Next. And so we looked to the study by Aditya et al., which examined the effects of cigarette smoke exposure on pulmonary gas exchange. They found that long-term exposure to cigarette smoke was associated with lower arterial PO2, higher arterial PCO2, and decreased diffusion capacity. Next. And so based on these findings, we theorized that PCO2 in the blood of smokers could reach critical levels more quickly, contributing to the quicker urge to take a subsequent breath after initiating a breath hold in comparison to their non-smoking counterparts. And so with the information provided by our background research on previous studies and the physiological mechanisms at play, we moved forward with the design of our study.
So how did we conduct our study? We had a total of 33 participants, both men and women, ages 18 to 40 years old. 17 were smokers and 16 were non-smokers. We defined a smoker as someone who smokes at least 10 traditional tobacco cigarettes per day. E-cigarettes like vape pens and e-cigars were not included. And a non-smoker is someone who never smoked, so no history of smoking. We did not include anyone who reported smoking fewer than 10 cigarettes a day because our main focus were at least 10 or more cigarettes a day to be considered a smoker. A survey was collected with their information that includes sex, height, weight, number of cigarettes smoked per day, and if there are any health conditions. For the sake of our experiment, we required all subjects to be healthy individuals, meaning no underlying health conditions or any current respiratory systems like allergies or any difficulty breathing that could affect their breath holding duration. If any participants were found to have any underlying issues, they will be excluded from the study. All information are strictly confidential. Next. After collecting all information, the subject is placed in a quiet environment, seated in an upright position. They are relaxed, taking slow, deep breaths, and they were instructed to rest for 5 minutes before the first breath hold. Next. In order to control the difference time passed since their last exposure to tobacco, smokers were asked to smoke 1 cigarette prior to the 5-minute rest. Next. After the 5-minute rest, using a timer, the first breath hold trial for both groups began. They were instructed to pinch their nose at the end of a single maximal inhalation and hold their breath. Once they can no longer do so, they will release the pinch on their nose and exhale. Using a stopwatch, breath holding time is recorded in seconds from the time they took their maximal inhalation to the time they exhale. Breath hold is repeated 3 times with 5-minute resting time in between trials. We used a Google spreadsheet to record the measurement for each trial and calculate the mean average. After recording all data, BHD average for smokers and non-smokers are calculated and compared along with the standard deviation by bar graph to better visualize whether there are any significant difference in breath holding time between the 2 groups. Next.
After collect the data, we got this graph. The graph shows the average breath holding time for participants who smoke 10 or more cigarettes per day in comparison to participants who are non-smokers. The blue bar represents the group of the participants who reported smoking 10 or more cigarettes per day, which has a population of 17. The red bar represents the group of participants who do not smoke, which has a population of 16. The vertical axis shows the combined average breath holding time of smokers versus non-smokers, ranging from 0 to 100 seconds. The mean for smokers is 50.37 seconds, whereas for non-smokers is 59.75 seconds. The standard deviation of the smokers is 9.72 seconds and of the non-smokers is 22 seconds.
So for our discussion, we concentrated mainly on a hypothesis that non-smokers can hold their breath for a longer period than smokers. So and our study concentrated, and it was found that there was not much difference in the breath holding time for smokers and for non-smokers, as there were not much significant gap between those 2 classes of people. Next slide, please. So we did concentrated on some other studies like which we did relate with our studies, such as Bodab, Sudan, and all. And for them, they did class smokers as those who have smoked at least 5 packs per year, and also their age gap were like 30 to 50 years. And we did have the same record in time for BHT as theirs. So they concentrated on taking the deep inhalation, and for subsequent period, then the subsequent breath holding time was taken. Then another study which we compared as ours with was Bodami and Tall. Both of these studies were from India. So with them, smokers were classed as individuals who have smoked 5 cigarettes a day for at least a year, and they concentrated only on male adults of 20 to 50 years, and also they used their same breath holding time. 2 of these studies have both similarities, so that's why we did a comparison with them. Next, please.
Our study from San Francisco Bay Area, smokers, anyone who self-reported smoking 10 or more cigarettes per day, healthy adult between the ages of 18 to 40. Breath hold duration measured in seconds from the end of participants' maximal inhalation until their first exhalation. Therefore, our study found not significant difference in breath holding time for healthy individuals 18 to 40 years, smokers and non-smokers. Next, please. However, in smokers, the exact effect leading to reduction of breath holding time is not clear. Gita Bhagavat et al. States that cigarette smoking contributes to increased level of blood PCO2 in comparison to non-smokers. In the PO2 is reduced or the PCO2 is increased in the blood, this can stimulate respiratory centers to increase activity. Suda et al. States that the main point of disruption, breath hold breaking point, is the increased level of PCO2. Breath hold break point is influenced by the stage at which alveolar partial pressure of oxygen has decreased to 75 to 50 millimeter mercury, and alveolar partial pressure and carbon dioxide has risen from 45 to 49 millimeter mercury. Next, tobacco smoking effect of the lung, such as lung cancer, chronic bronchitis, emphysema. And the end, no smoking, beautiful life. Thank you.